Good afternoon, children. Welcome to Vivid Botany. We have a question about the bonds here. A very good question, especially the question is framed on the bonds. You see here, there are two types of bonds are given. One is glycosidic bond and the other is peptide bond. Let me explain you the two bonds very clearly. In fact, glycosidic bond, peptide bond, both are covalent bonds. But a glycosidic bond is generally present in carbohydrates and a peptide bond is present in amino acids that make up the proteins. So a peptide bond is present between amino acids to form proteins. I will give you examples also. Whereas a glycosidic bond is that it is present between monosaccharides generally, that is units of the carbohydrates. Now let me explain you that a glycosidic bond can join two monosaccharides to form a disaccharide. A glycosidic bond can join two monosaccharides. Let me explain you with example also here. A glycosidic bond can join two monosaccharides to form a disaccharide. So what is the monosaccharides and what is that disaccharide? What they are exactly? See here, two monosaccharides can be taken as the glucose and fructose. Glucose and fructose both are monosaccharides. They join together and they form a disaccharide, that is sucrose. Many polysaccharides such as starch, cellulose, glycogen, chitin, they contain monosaccharides units joined by glycosidic bonds. So we can understand the glycosidic bonds are generally present in the polysaccharides, that is carbohydrates. Another example I can give you, that is inulin, which is made up of 1 glucose unit and 2 to 60 fructose residues. So we can conclude that here glycosidic bond is present concerned only about the polysaccharides, only about the carbohydrates. Coming to the peptide bond. Peptide bond is formed, I wrote here, present between amino acids to form proteins. So proteins are called as polypeptides. Every protein is polypeptide. So how this peptide bond is formed? Let me explain you that a peptide bond is always formed in between the carboxylic acid of the first amino acid and amine group of the other amino acid. So let me explain you about the peptide bond. So let me see a diagram here. This is amine, NH2. Then we have the alpha carbon, hydrogen, R1. Then it is having the carboxylic acid, C double bond O, COH. So this is one amino acid having the R1 group. Now this amino acid will form a peptide bond with other amino acid. The formation of peptide bond is quite interesting. Remember here is that carboxylic acid of first amino acid and amine group of second amino acid will form the peptide bond by loss of one water molecule. So let me see here that this is alpha carbon, hydrogen, and this is the alkyl group R2, and again the carboxylic acid. C double bond O and OH. Now, these two amino acids will form one peptide bond. So, this is again NH, H, alpha carbon, hydrogen with R1 alkyl group, C, CO, and again we find here HN, NH, and again alpha carbon, CH, R2, C double bond O, and OH. So, this bond which I am marking here is called as the peptide bond. So, peptide bond and even glycosidic bond both are dehydration bonds and both are covalent bonds, but peptide bonds are formed only in between amino acids, whereas glycosidic bonds are formed in between the sugar molecules, maybe monosaccharide or disaccharide units to form the polysaccharides. So, hope you have understood. Once again I am reiterating that a peptide bond is always formed only to form the proteins in between amino acids, only to form proteins, but not in carbohydrates or in lipids the peptide bonds are present, whereas the glycosidic bond is present in between the saccharides, can be monosaccharides, disaccharides to form the polysaccharides. So that is the main difference here. Let me explain the question once again here. Identify the substances having glycosidic bond and peptide bond respectively in their structure. First one you see chitin. Chitin is a carbohydrate. So here glycosidic bond is present, and cholesterol is lipid. So in this lipid peptide bond is absent. Second one you see glycerol. IUPAC name is trihydroxypropane. So in this glycerol only hydrogen bond is present, not the glycosidic bond. What is the difference in between hydrogen bond and the glycosidic bond? A hydrogen bond is formed between hydrogen and another atom that is fully electronegative atom, whereas a glycosidic bond is formed in between the saccharide units, the sugar units. So glycerol contains only hydrogen bonds, so it's not glycosidic bond. It's also wrong. Cellulose contains, yes, glycosidic bond, but lecithin is phospholipid. Lecithin is basically lipid. So a lipid does not contain the peptide bond. So third option also wrong. Let us come to the fourth one. Inulin. I wrote here inulin is made up of, one glucose unit and 2 to 60 fructose residues. So inulin definitely contains the glycosidic bond, and insulin is a hormone and basically it is a protein. So protein contains the peptide bond. So option 4 is right for this question. So remember always protein will have different forms. A protein can be antigen, protein can be an antibody, protein can be a hormone, a protein associates with pigments, a protein can be toxin, protein can be a drug, right? So protein have different morphs, different forms to perform different functions. We see that all enzymes are proteins except the ribozyme. So proteins will have different functions with different forms. So every protein will have chain of amino acids that are bound by peptide bonds. So here the fourth option is the very much correct. The question is very much tricky here. You should know first of all which one is the protein here, which one is lipid or carbohydrate here, and you should know whether peptide bonds are present in lipids or carbohydrates or not. They are absent in lipids and carbohydrates. That you should know very clearly. And you should know that glycosidic bonds are present in lipids and carbohydrates. When you know all these factors very clearly, then only you can tick the right option correctly. That is a problem here. So this is question basically framed very nice question based upon only the bonds and the type of bonds. So merely if you practice the diagrams of NCERT, it's not enough. You should know what is the type of bond, what is the hydrogen bond, what is peptide bond, what is a covalent bond, what is ionic bond, what is the difference in between glycosidic bond and peptide bond. You know both are dehydration bonds, both are covalent bonds, but peptide bonds are absent in lipids and carbohydrates and glycosidic bonds are absent in proteins. So students should know this difference, then only he can do the question very much correctly. So I expect my explanation is going well and it is reaching your levels of understanding, and please don't forget to subscribe my channel. We will be explaining all the questions one after the other, like we are going now 2020. Earlier I have explained 2021 NEET questions in my Vivid Botany channel. Please do subscribe and see all the explanations and drop your comments in the comment box, and you can WhatsApp me also, you can Instagram me also if any doubts are there. We will discuss them in our next videos. Thank you.